I'd like to go soon. All right, anyway, we're going to be doing cells as soon as we get a chance here. As far as the cell...
I think the PowerPoint is frozen.
It's all right. We'll make it work. Here's the kind of the list of things we'll talk about. Characteristics in common with all cells. Prokaryotes versus eukaryotes are two main categories of cells. Plant and animal cells. All the different cell parts. And then how things get in and out of cells with transport. So all cells contain, there's four things. Whether we're talking about prokaryotes, eukaryotes, any kind of cell, they're all going to have four things in common. Okay, they're all going to have DNA. They're all going to have ribosomes in them. And if we think about these a little bit, that makes sense. Because DNA is the code to make proteins. And what does the manufacturing of proteins? Well, ribosomes does that. So you have every living thing, whether you're talking a bacteria, like a prokaryote, or a eukaryotic cell, they're going to have DNA, they're going to have ribosomes, they're all going to be surrounded by a cell membrane, and they're all going to have cytoplasm, some sort of like cellular soup holding in nutrients and things that that cell might need.
Mr. Lyons, just to remind them that on the keystone, if you see plasma membrane is the same as the cell membrane.
Good point, good point. All right, differences between prokaryotes and eukaryotes. If you can think, pro rhymes with no. Prokaryotes have no nucleus. Okay, that's their major difference. Whereas eukaryotes, like humans, plants, animals, fungus, we all have a nucleus. We all have those membrane-bound cell parts, the mitochondria, the ER, the nucleus, all those different parts. So they're part of eukaryotic cells. Specifically looking at those eukaryotes, we can split them into two groups, plant cells and animal cells. As far as animal cells go, a couple key characteristics, which I think we have on our next. Yeah, okay. So as far as, here's prokaryotes and eukaryotes again. Again, main difference, no nucleus to nucleus. Some of the other differences there are good too. Here's the things all cells have. The plant cells, this is kind of some important things here that plant cells have that animal cells don't. Plant cells are surrounded by a cell wall. They've got chloroplasts. The process of photosynthesis happens here. If we had chloroplasts, you wouldn't go to lunch. You would just go stand outside and suck in sunlight. It doesn't really work that way. So, because we have mitochondria, not chloroplasts. So we've got cell wall, chloroplast, and a central vacuole, which is basically like a giant bag inside of the cell that stores water. And it kind of keeps pressure pushing out on that cell wall so the plant has shape and has structure to it. Because think about a healthy plant, if we're talking about something herbaceous, something with like a soft stem, it stands up in the air. It's pretty rigid. But if you stop watering it, what happens? It starts to wilt and kind of goes limp. Well, it's because this vacuole collapses inside of the cell because there's no water in it. And then the cell wall folds in on itself, and now you can't stack those bricks. The plant cells kind of look like bricks. You can't stack them on top of each other, so then they kind of tip over. The fact that there is a cell wall is what gives those cells their square shape. As far as animal cells, we don't have that on this slide, but animal cells do have one thing that plants don't, and that's lysosomes. So if you see something about something an animal cell has that plants don't typically have a lysosome in them. That's something specific to animals. Same thing with a flagella. Like on the end of a sperm cell, they have that tail. Plant cells don't have those. They don't have any kind of mobility to any of their cells. All right, looking at some specific cell parts, okay? So these are going to be ones in common, at least most of the way down here, with all types of cells, or to all types of eukaryotic cells, both plant and animal cells. So mighty mitochondria, think they're mighty, they're strong, they're the powerhouse, they make ATP. If you can think ATP, energy, okay? ATP is energy for our cells. So when you eat your food, and we'll talk about this a little bit more with the respiration and the energy unit next, the food that you're eating is broken down to make this chemical energy, the ATP. And most of that process happens in the mitochondria. We already mentioned ribosomes, protein synthesis, that's where the proteins are made. they get the code from the DNA, they make the protein, then that protein is produced, it's shipped down the ER, it's sort of like a little highway system to move those proteins, and then they get sent over here to the Golgi where they get packaged into these nice little vesicles so that they can get sent out of the cell. And that vesicle that they're made into, that they're packaged in, is actually the exact same material as the cell membrane. So when this vesicle hits the cell membrane, it actually becomes part of the cell membrane, and whatever's in it can leave the cell. So besides plant cells having like that vacuole that pushes out to kind of give the cell shape, we have some of that too. It's our cytoskeleton. It's microtubules and microfilaments. They're basically like a scaffold inside of the cell that kind of pushes and holds that cell in place. And then we have the nucleus. It's our control center. It has the DNA in it. Basically, it makes sure that all the functions of the cell are being done correctly. Within that has the nucleolus, which is its job is to make ribosomes. And then lastly, typically only in plant cells, are our lysosomes. Like a little Pac-Man going around and getting rid of trash. Digests all the waste in cells, things that our cells really don't need anymore. Old organelles, have a bacteria or something happens to get into the cell, they're able to kind of digest that and get rid of it. Okay, so specifically looking at the cell membrane itself, what does it do? It's our gatekeeper. It's like the bouncer of the cell. It regulates what comes in and out. It makes sure that the food that we need, the nutrients that we need to get into the cell, those things can get in, and there's different ways we'll talk about in the next one, how those things get into the cell. But it also needs to keep out things that we don't want, like bacteria and viruses and anything that could harm our cells. So it acts as a gatekeeper. We mentioned this, plasma membrane, cell membrane, interchangeable words, they mean the same thing. Now, as far as the structure of the cell membrane, we say that it's selectively permeable, but that it is a phospholipid bilayer. So what does that look like? Okay, so here you have your phosphate heads, which they are polar, and you have your lipid tails, which are nonpolar, and they kind of repel the water away, repel each other away, and they make this nice little boundary around our cells. It's sort of semi-fluid, like it can, sort of like those stress balls, you can squeeze it and it changes shape, but you let it go, it kind of goes back to normal again. Our cells are kind of that same setup in the way that this membrane works. Okay, so you have this going the whole way around your cell. The two main components are lipids and proteins, but there's also some cholesterol that's there. I mentioned it's sort of stress ball-like. Sometimes they refer to that as this fluid mosaic. It's a bunch of parts put together. That's where the mosaic piece comes into play. Fluid because it's not quite really a solid, because it's not, all of the pieces aren't held together by bonds. They're just kind of held together by sort of attracting and repelling things that are around them. And lastly, from this section, transport. How do we get stuff in and out of this cell? Through the cell membrane. So you have passive transport and active transport. I like to, and I'm going to piggyback on her analogy of the hill, I like to think about this as like a guy with a ball on a hill. If he's standing at the top of the ball and he lets go, does he have to use any energy to make that ball go down the hill? No. It naturally rolls down the hill on its own. So this is like passive transport. If I'm at the bottom of the hill and I have a ball and I want to get it to the top, I'm going to have to throw it, roll it, push it, carry it, do something that requires energy to get that ball to the top of the hill. So that's active transport. The hill itself represents our concentration gradient, okay? Going from either an area of high concentration to low or from low to high, moving against that natural movement of molecules, that natural gradient that they want to travel in. So passive transport, high to low concentration, room from high on the hill to low on the hill, no energy required. There's three types of that: diffusion, osmosis, facilitated diffusion. Diffusion's real easy, just gases pass in and out of the membrane. There's nothing, like they fit, I've erased the picture already, But in between the phospholipids, they just sort of meander their way in and out. Osmosis, same general idea, but with water. Okay, they use little protein channels to pass the water in and out of cells. But this one, I want to make sure we understand what the word facilitate means, because it's easy if we know what the word means. Facilitated diffusion, it's the same as diffusion, except facilitated, a synonym for that is the word help. So it's diffusion with help. Well, what's doing the helping? Proteins are doing the helping. They basically, instead of squeezing real slow through a really skinny door, we're opening up a really wide door so lots of molecules can get in at once. Okay, that's kind of the benefit to this. Sugar is something, because we need lots of sugar coming into our cells. Sugar can get in through diffusion, but it's real slow. Okay, it's like squeezing through a real tight space. Whereas with facilitated diffusion, we can open a really big door and a lot of sugar can rush into the cell at one time.
Can I jump in here for a second? Because I have some amazing hand gestures to help with these proteins. Okay, so in facilitated diffusion, there are 3 types of proteins. I don't know if you need to know this for the keystone, but it's fun. The names of the proteins are the channel protein, the carrier protein, and the gate protein. The channel protein is basically just making a little tunnel for the molecules to go through. So put your hands like this, and you can go shh, shh, shh. That's the channel protein. The carrier protein carries the molecules across the membrane. So it kind of puts them in a little basket and drops them on the other side. A little faster though. And then my favorite protein, the gate protein, requires, come on Camden, you can do this with me, requires a gate to open, and then once the gate is open, the molecules can come through. I'm guessing Mrs. Contras's students never did this, huh? No. Okay, so my sound effect for that goes ding dong, eek, and the gate opens. Just thought I would share that with you. Should we talk about hypo, hyper, and isotonic? So out of the frame.
Yeah, it's okay. So when we talk about osmosis, we're trying to figure out, are cells going to take in water and swell up and get bigger? Are they going to lose water and shrink and get smaller? Or based on the environment that they're in, they're not going to change at all? So we have hypertonic environments, would be a hypotonic environment, or an isotonic environment. Here's what looks different about these. If you take a cell and put it in a hypertonic environment, there is more water inside of the cell than in its environment, and water is going to leave. It's going to shrink and get smaller. This would be like if you go swimming in the ocean for a while and you get out and your fingers are all like wrinkled up. It's a hypertonic environment. Water has left your cells. You have shrunken and gotten smaller. Easy way, the way I remember this, I think of an E looks like a shriveled up O. And an O is nice and big and swollen. So hypotonic, you're laughing at me, I'll laugh at your hand gestures. Hypotonic means water's coming in. Okay, so you're putting cells in an environment where they want to take in more water, they're going to swell up and get bigger. You guys probably did the egg lab at some point. You know, in the vinegar and the water, the egg swelled and got bigger because they were hypo environments. And in the syrup, it lost water, so it shriveled, so it was in a hypertonic environment. Isotonic, same amount of water coming in as going out. It's just you're putting that cell in an environment that has the exact same concentration of water and solutes as the cell does. And I think oftentimes, like the book describes the movement for these in terms of the movement of solutes, which I think makes it really hard, because osmosis is the diffusion of water. So why don't we talk about it in terms of water? So hypertonic, water leaves. Hypotonic, water comes in. Isotonic, no change in size. Active transport, moving on. Active, we're going from the bottom of the hill to the top of the hill, so we're moving up that concentration gradient against the natural movement these molecules want to move in. So that to do that requires energy. Remember, ATP, energy, has kind of a little rhyme to it. 3 types of active transport. You have endocytosis, think EN for enter, Exocytosis, think EX for exit. Okay, so this is big things like proteins coming into the cell. or out of the cell. And then you have pumps like a proton pump or the sodium-potassium pump that actually have kind of like a trigger mechanism that works, that actually pumps one thing into the cell and one thing out. It pumps sodium out of the cell and potassium into the cell. And again, that has required ATP. So it's a pump. So it's like if you got water in your basement, it's not just going to go away on its own because the water around your house is higher. So you have to pump it out of the basement.
On a Keystone exam, if there is a random picture of your lipid bilayer, and then let's just say there's a protein in it, and it asks you what kind of transport is going on, if you see an ATP in a flashy cloud, it doesn't matter what's going on in this picture. As long as you see this ATP, you know it's active transport. I think most of the time there's some kind of random picture of a lipid bilayer. Look for that ATP. If you see it, it's active transport. If you don't, for the most part, it's going to be passive.